today I'm going to be talking about phagocytosis. Now, first off, phagocytosis is the process in which a phagocyte, so this could be a neutrophile, a macrophage, a dendritic cell, any one of those cells that essentially is a process in which they engulf a foreign microbe within your body. So this could be a microbe, a bacterial cell, some sort of debris, anything that is really foreign to the phagocyte, they will ingest and it'll go through this process called phagocytosis. So the first step in phagocytosis, what really gets things started, is due to chemotaxis, so the attraction to chemical stimuli. This is something that phagocytes rely on, and this could be through C5a, C3a, I believe, cytokines, stuff like that. So anything that will attract the phagocyte through chemical stimuli is what gets this process of phagocytosis started. And then the next step would be adherence. So the main way that they adhere is through toll-like receptors, but they can also use opsinin receptors. But for the sake of this video, I'm really just going to focus on TLRs. So the toll-like receptors will bind to something called a pathogen-associated molecular pattern, a PAMP. So TLR will bind to PAMP. Essentially, this is what allows the microbe to adhere, but it's also important to know that TLRs aren't just specific for one chemical or physical structure within a microbe. A lot of TLRs have, or within a microbe, a lot of TLRs have or are specific for things like flagella, lipopolysaccharides, peptidoglycan, stuff like that. So depending on the TLR and depending on the microbe, they may or may not bind. And for example, some bacteria are encapsulated, so they have a capsule, meaning that the phagocyte struggles to adhere to it in the first place. Something called opsonization can happen. Opsonization is essentially when they take or essentially when they cover the encapsulated bacteria or microbe, whatever, with anything that the phagocyte can bind to. So that's called opsonization. So there's a workaround to get around the fact that capsules cannot be, or they have trouble getting adhered to. So the next step is ingestion. Ingestion happens when the phagocyte engulfs the microbe and internalizes it within the cell, forms something called a phagosome. And then after a phagosome forms, it's a membrane bound sac, by the way. After a phagosome forms, a phagolysosome will form, which is essentially the fusion of the lysosome and the phagosome. Lysosomes will then go into digestion, and then these certain enzymes that are breaking down the microbe, whether that be nuclease, lipase, lysozymes, anything like that, any of those enzymes that will break down a microbe, start to fuse and digest the microbe within the phagolysosome. Finally, exocytosis will happen, which is when the remnants or the dead cell, the debris or the parts of the dead cell are essentially expelled from the cell, and then the phagocyte will move on to engulf another cell. So it's a very intuitive process when you come to think about it. So chemotaxis, adherence, ingestion, phagolysosome formation, digestion, exocytosis. One thing that I forgot to point out, some kind of like a fun fact, is the phagolysosome has a pH of around 4 to 5, so it's already pretty acidic. This is of course done on purpose so that the environment, or that the phagolysosome for the microbe is already starts to break it down, or the fact that it's acidic makes it difficult for the microbe to thrive in that pH. So that's something I forgot to point out, but yeah, and then I drew it out right here. So simple, super simple, quick and easy topic, and I hope you guys all learned something.